All right. Good evening, everyone in Cleveland. Thank you very much for joining us for this very exciting session of BrainX Community Live, the last one for 2021, this one being for December 2021. And we have some very exciting speakers today to present some really interesting research work and their experience with AI and biomarkers in the field of cancer, addressing some of the healthcare disparities and so much more. But before we get to that, I just want to introduce BrainX Community for those who are joining us for the first time. We started this community more than 3 years ago with the idea of machine learning and healthcare for good, and so that we can share a lot of scientific material, create a platform for collaboration, for data sharing, and for enrichment for everyone. So we have had 3 very successful years. Our community has grown to be 3,000 members strong and continues to grow every single day. Very proud to announce that just on our LinkedIn group, which is a very active group where we share a lot of scientific information, now we have BrainX community members crossing 2,000 in number. And this is from all walks of life, from engineering, from healthcare, from students to full professors. A lot of people have joined our group on LinkedIn, so I would highly suggest, if you're not a member yet, please join either through our webpage or through the LinkedIn group, or both. A few resources for you that I'll go over are our websites, the brainxai.org or brainxcommunity.com. And over here, we have our monthly live sessions. So if you go to our website, what you'll find is that we have renovated it. It's a completely new platform, way more interactive, and providing you all the resources readily at your fingertip. We have the homepage where we have the updates, where you can find all the different information which is readily available to you in a manner where you can either look at that particular update, or you can view all to look at some of the prior updates related to that particular section. So amongst the sections, we have the Connect section, where we have presentations from our previous BrainX Community Live sessions, which are posted over there, or the ones that are going to be coming up, just like this one. Once we finish, it will be there on our YouTube video. It will be accessible through our website, and you can readily access it right over there. Another one that we have, other than the Connect section, is to go to the YouTube channel. All the prior presentations are posted over there. You can subscribe our YouTube channel, so as soon as the presentation is posted, you can view it readily over there. As I mentioned, our very active LinkedIn group, now crossing 2,000 plus members. Please join that. Please help us spread the word. And then the Learn section, where we post some of the latest curated publications. So you can go to this particular section and look at which are some of the real interesting publications that have come across in different areas. They might be in medical journals, they may be in the engineering journals. We curate those articles for you based on their importance. We provide you direct links to the journals where they have been published. We categorize those for you so that if you're interested in a particular specialty, you can search them, you can filter them by that particular specialty, and you can also filter them by the date or the year. We do have a journal section so that you can look at some of the key journals which are specifically focused for publications related to AI and healthcare. That way, you don't have to go to multiple places. In one particular place, you can find all this information. And then books, a lot of books that are getting published over here, they are listed over here. So through the Learn section, you can filter for books, and you can find interesting books which will help expand your knowledge base. And something that everybody is looking for are open source data sets. So we are very proud that we have one of the largest listing of links to open source data sets to fulfill your needs. So if you go to the data section on brainxcommunity.com or brainxai.org, you can go and find all these open source data sets, once again curated for you, once again curated based on different specialties, and with some information about what's in the data set with direct links to those data sets readily available. So you can start working on your next interesting model. And we all are trying to learn, and we are trying to learn from some of the experiences of some of the greats in this area. So we have both clinicians and engineers and innovators joining us for our podcast series called BrainX Talks. The one that we just recently completed with Dr. Suchi Saria is posted. We have our own Apple Podcast, Google Podcast, and Spotify channel with the name BrainX Talks. So please subscribe that and get the latest podcast. We have also featured Professor Anant Madhabushi on this previously. So please listen to his views. Again, a leader in this field, and has established the CCI-PID as a world-class group, and a lot of members from his group and his team joining us today. Another thing that many people look for are meetings and conferences where people can interact, where people have the opportunity to present their papers. So again, these are available through the conference section, readily available through the meetings or conferences area. You can register directly through the links that are available on our website and attend those. AI in Oncology Symposium was very successfully held by, again, the group of presenters who are going to be speaking to us today. So again, if you're looking for the next best meetings to go to, please look at this section. And today, I'm going to hand it over to Dr. Rakesh Shiradkar, who's the research assistant professor for biomedical engineering and at the Center for Computational Imaging and Personalized Diagnostics at the Case School of Engineering, Case Western Reserve University, and he's going to help moderate this exciting session. So with that, I'm going to hand it over to Rakesh. Over to you.
Thanks, Piyush, for that intro on BrainX and also the introduction for this talk. Well, good evening, everyone. So today we'll be presenting some of the work that we've been doing at our Center for Computational Imaging and Personalized Diagnostics on AI and computational pathology in identifying population-specific biomarkers, specifically in the context of breast and oropharyngeal cancers. So this talk, I'll mostly be moderating the talk, but the actual presenters are Haojia Lee. She's a PhD candidate in the lab. And we also have Dr. Koyuncu, who is a research associate in our lab. So, yeah. So as you all must be aware, cancer is a big burden globally, and especially in 2021, the NIH estimates 1.9 million new cases and about 600,000 cancer-related deaths. But something of interest to note is that certain population groups are disproportionately affected by cancer incidence and cancer-related deaths. So here is a chart that we have, and you can see that Black men and women, they have the highest rate of cancer death, and especially Black men, as you can see, have a higher incidence of cancer among all other racial and ethnic groups. So there have been several studies that several groups have focused on, and what they've come across is that the factors contributing to this disparity is mainly an interplay of several factors, including social determinants of health, biology, genetics, diet, physical activity, etc., so on and so forth. Then coming to AI and computational pathology. So histopathology, as most of us should be aware, that is the gold standard for most cancers. It's used to establish the grade and the aggressiveness of the cancer. So mostly tissue is biopsied, it's H&E stained generally, or with other immunohistochemistry stains observed under a microscope and diagnosed. And AI and computational pathology has enabled automated characterization and high throughput extraction of some of these features, including the arrangement of nuclei or glands, the shape, the texture of these glands, quantifying those automatically. And these have been used to build several diagnostic and prognostic models. So our group and several others have shown this to be very promising in several cancer sites, including breast, prostate, lung, kidney, eye disease, so on and so forth. So coming to the topic of health disparities. So although there's an interplay of several factors, one of the most important factors is biological basis for this cancer disparity. So several high-profile papers have shown that there are specific genetic mutations associated with certain population groups that may be associated with the observed disparity. However, the current nomograms that we have and the predictive and prognostic models that we have largely don't account for population-specific differences, and there's a one-size-fits-all nomogram. So this may not be as efficient in identifying who might be, benefiting from a certain therapy, or who might, result in better outcomes. So accounting for population-specific differences may result in better prognosis. So as a case in point, we've recently published a paper in clinical cancer research where we showed that a certain signature derived of stromal morphology was prognostic of prostate cancer recurrence in African-American patients. And the same signature, when applied to a Caucasian cohort, was not as prognostic. So our hypothesis towards this research is that population-specific differences, as evidenced at the genetic level, may be reflected at histopathologic scale, and AI and computational imaging techniques can help characterize some of these population-specific biomarkers, which can then help build targeted models towards specific populations, which can result in improved outcomes. So in the context of this talk, we'll be specifically looking at breast and oropharyngeal cancers, some of the preliminary research that we've been doing in this area. So Haojia will present her work focused on the role of computational pathology and AI in characterizing breast cancer differences between South Asian and North American women. Haojia, go ahead.
Hi, everyone. So thanks for attending the talk. So I'm a PhD candidate in CCIPD. So I'm going to talk about the racial disparity in the context of breast cancer. So here is some background. So breast cancer has become the most commonly diagnosed cancer worldwide in 2021, accounting for 12% of the all new diagnosed cancer cases globally. Fortunately, the mortality rate for breast cancer has declined in recent years due to the increased emphasis on early detection of the breast cancer using the mammograph screening and the administration of the more effective treatments. However, the increase in the breast cancer patient survival mainly happens for Caucasian population. There is still a huge racial disparity and also ethnicity disparity in breast cancer incidence and mortality rate across US. So at the first figure on the right shows, you can see that for non-Hispanic Black patients, they have the highest mortality rate compared to other racial groups in the US. And also the racial disparity exists in terms of molecular subtypes, as the figure on the rightmost shows. So the molecular subtype is determined based on the hormone receptor status, as well as the human epidermal growth factor receptor 2. So the basal subtype is a subtype which has the worst prognosis. So as you can see here, the non-Hispanic Black patients has the highest proportion of the basal subtype of breast cancer. So besides African American patients, multiple studies have also shown that the South Asian patients are more likely to be diagnosed with the more advanced stage breast cancer compared to the Caucasian patients. In addition, the proportion of basal subtype for South Asian patients is also higher than other overall than other regions, than the patients in other regions in the world. So as Rakesh mentioned in previous slides, the potential risk factor could be the socioeconomic status and etc. And more importantly, studies have also demonstrated the variance in the biological and genetic difference between the tumors for the different racial groups. So however, the existing clinical models for recurrence risk prediction for breast cancer didn't account for the population-based variation. For example, the Oncotype DS, which is one of the most widely applied gene tests for recurrence risk assessment for breast cancer, is less accurate in African American patients compared to the Caucasian patients. So the potential reason could be that the test was predominantly developed on Caucasian patients, which result in the failure of taking into consideration of the genetic variance between the African American patients and Caucasian patients. So in this study, we focus on the lymph node negative and estrogen positive invasive breast cancer. So estrogen receptor positive breast cancer is a breast cancer which grew in response of the estrogen. And the administration of the hormone therapy could block the estrogen receptor and then slow down the growth of the breast cancer. So the lymph node negative breast cancer indicate that the breast tumor is still confined within the breast tissue and has not migrated into the adjacent lymph nodes. So ER positive and RN negative breast cancer is the early stage cancer which typically have favorable outcome. So the standard treatment is breast conserving surgery plus the adjuvant hormone therapy. The adjuvant chemotherapy has also been demonstrated to be able to increase the survival. However, not all the patients derive significant benefit from the chemotherapy. So it is important to identify the low risk ER positive lymph node negative patients so they can safely omit this chemotherapy. The quantitative tumor histomorphometric features have been demonstrated to be prognostic of ER positive lymph node negative breast cancer. However, those models were mostly trained on the Caucasian patients. So motivated by the racial disparity discussed in previous slide, in this study, we investigate the capacity of the digitally measured population-specific phenotype of the tumor nuclei to create the improved population-specific model for breast cancer prognosis. So here is our workflow. First, we collect the H&E stand slide imaging from the South Asian population, North American population, and all the slides were digitized as 40x. So we have pathologists to annotate the breast tumor on the whole set imaging. And then we use a deep learning model to segment the tumor nuclei. Following that, we extract 241 different nuclear features from the tumor. Finally, we train the population-specific model based on those features to predict the cancer outcome. So this is some details regarding the nuclear feature extraction. So we extract 5 different categories of features. First, we extract the small tumor tiles from within the tumor region annotated by the pathologist. We extract the shape feature, which capture the nuclear boundary properties such as irregularity and smoothness of the nuclei. And also we extract the cell orientation disorder features, which is used to measure the disorder degree of the nuclear orientation. And in addition, we look at the global graph feature, which describing the global spatial distribution of nuclei, as well as the cell cluster graph feature, which describe the local spatial distribution of the nuclei. And finally, we look at the Harlick texture feature, which characterize a chromosome pattern within each nucleus. So this is how we construct the population-specific model. Following the feature extraction, we use the Cox proportional Heather regression model regularized by ElasticNet to associate the extract nuclear feature and the disease-free survival. So disease-free survival is the outcome or the endpoint we used here, which is the time interval from the cancer diagnosis to recurrence or distance metastasis or death. So this the Cox regression model is able to identify the features that are mostly associated with disease-free survival and also assign each feature a coefficient. So subsequently, we calculate the continuous risk score by summing up the feature values weighted by the corresponding coefficients. After that, we apply a threshold to distribute the patients into high risk score group or low risk score group. So a high risk score indicates that a higher predicted likelihood of recurrence by the model. So this is the data we collect. For the South Asian patients, we have 69 patients collected from Tata Memorial Center in India. And we also have 121 patients collected from the University Hospital. So all of those patients is HER2 negative, and the median disease-free survival for South Asian patients is 6 years, and South American patients 6 years, and South Asian patients is 7 years. So we built 3 different models, respectively, with the patients, with the South Asian patients, North American patients, and a combination of South Asian and North American patients. So then we respectively validate those 3 models on the South Asian population. So as you can see here, for the 3 models trained with different population, for the first model, trained with South Asian patients, we call it South Asian model. Most of the top prognostic feature identified are texture feature. However, for the North American model and the South Asian plus North American model, the prognostic feature are shape feature and orientation disorder features. And also another interesting thing we can notice here is when we validate the South American model on the, validate the North American model on the South Asian population, the KM curves is kind of flipped in terms that the high risk population has like almost significantly favorable outcome compared to the low risk group. So this indicates that in terms of this model, the high risk South Asian population behave more like low risk North American population, and the low risk South Asian population behave more like high risk North American population. And also you can see here, the model is only prognostic on the South Asian population when it is also trained with South Asian population. So since we notice this flipped KM curves here, so we're interested to see like how the shape features are different between those 2 racial groups. So here is a plot of the distribution of the 2 populations with respect to the 3 top shape features. So this the first top shape feature is the median standard deviation of the distance. So the distance here is distance from the nuclear centroid to the points at the nuclear boundary. So if the standard deviation is 0, then it means that the nuclei is in a perfect circle. So the higher standard deviation indicates the higher diverted, the higher the shape is diverted from the circular morphology. And another two features are related to the Fourier descriptor. The Fourier descriptor is able to describe the complexity of the nuclear boundary. So however, we want to make sure that this difference we observed is really from the difference in the population instead of the difference in the other confounding variables such as tumor grade. So so we already know that in South Asian population, more patients are in high grade compared to the North American patients, where most of the patients are intermediate grade. So in order to dig into this aspect, we perform additional experiments here. So first we look at this distribution of the patients with respect to the different grades. You can see there's a separation. So we want to make sure that the separation we observed here is not driven by the separation among the different grades. So then we separately plot out the distribution between the populations within like each histological grade. As you can see, we can still able to see the a certain level of separation when the patients have same tumor grade. Okay, so this is a visualization of some top prognostic features of South Asian patients. So this top 3 feature, hardic texture feature. So as you can see, the higher the patients in high risk have the higher like intensity variance. So that indicates that the high risk patient, high risk South Asian patients has a higher heterogeneous chromosome pattern within the nucleus. So okay, so the conclusion is the nuclear histomorphometry feature different between the breast cancer patients from South Asian and North American, and also the prognostic capability of the models for the South Asian patients can be significantly improved by taking into account the population-specific information. So for the future work, since in the previous slide, we also demonstrate the disparity between the Caucasian patients and American patients. However, in this study, we didn't separately, like, we kind of groups them together as North American patients. So in future work, we will separately looking at the 2 racial group, and then we will have 3 different racial groups of African American, Indian, and Caucasian. And also we will separately investigate the histomorphometric feature of the nuclei in different types and different regions. We look at the lymphocyte in epithelia region, lymphocyte in stroma, and also the tumor nuclei in epithelia, and etc. Yeah, so I think that's my part. I will hand over to ZHANG. Okay, so I will stop sharing.
Yeah, let me share my screen. So can you see my screen?
Yes, it's good. Yeah.
So first of all, thank you for providing me this opportunity to present my study. I'm Koyuncu, a researcher at CCRPT. And today I'm going to talk about the projects where we implement quantitative imaging biomarkers aiming for prognosticating the survival of HPV-related oropharyngeal squamous cell carcinoma patient. First, I will briefly describe the HPV-related oropharyngeal squamous cell carcinoma, and then role of multinucleated tumor cells in oropharyngeal cancer. And then I will explain how we approach the problem of prognostication using our AI-driven technique. And then I will talk about the population-specific differences in tumor biology of oropharyngeal patients in terms of our imaging feature. So oropharyngeal squamous cell carcinoma is one of the most common head and neck cancer type. And the latest staging system, that is AJCC 8th edition, that separate the staging criteria for HPV-related and HPV-independent patients, because studies show that HPV-related patients have favorable survival compared to HPV-independent one. So in our study, we just focus on HPV-related oropharyngeal patients. And the identification of HPV status have been performed by immunohistochemistry for P16 gene, because expression of P16 positivity is highly correlated with HPV infection in head and neck cancer. So the current gold standard use TNM staging along with some clinical parameters, such as smoking status, to predict the aggressiveness of cancer. But some studies demonstrate that the current pipeline guidelines do not work well for risk stratification. For instance, Gupta et al. Demonstrate that there are no significant differences in survival time of patients categorized as stage 1 and stage 2 by AJCC 8th edition system. And similar studies, another study found similar results for stage 2 and stage 3 patients in a large cohort. So one reason could be that the parameters utilized by the system, by the guideline, do not capture any biological characteristics of tumor, which may be relevant for treatment sensitivity. So to do so, people focus on finding single or multi-gene prognostic signature. However, these studies have some limitations. For example, most of the genomic analysis, genomic biomarkers, they were not verified or validated in independent institute or independent data set. And genomic tests are expensive and they are tissue distractive as well. So we cannot, it is not possible to apply the same genomic test on the same sample. Because of that, quantitative imaging biomarkers have become popular recently. The tissue morphology and architecture within the tumor microenvironment is reflective of tumor aggressiveness, and they carry a lot of prognostic information. So based on this fact or hypothesis, quantitative imaging biomarkers aim for extracting features from related to tumor morphology or architecture. However, many of these biomarkers do not take racial disparities into account. And there are several studies that show there is a significant racial disparities in many cancer types. In the context of oropharyngeal cancer, one study compared the mutational profile of African American and Caucasian American patients. And they found that African American tumors were found 1.4 times more likely to show loss of CDKN2A gene, and that specific gene was associated with malignant progression in oropharyngeal cancer. And in the same study, they also revealed that AA tumors were found 57% less likely to have loss of SCYA3 gene. And that gene plays an important role in lymph node metastasis in oropharyngeal cancer. So in this talk, first I will talk about, I will discuss our AI-driven solution that aims for prognosticating the survival of patients. And then I will talk about how we assess the population-specific differences using our biomarker. So one previous study conducted by our pathologist collaborator, Dr. Lewis, he's an head and neck pathologist. He previously proposed that tumor multinucleation is correlated with patient, cancer aggressiveness. So multinucleation is a kind of cancerous cell. There's a cancerous cell that contains multiple nuclei inside. And they propose that frequency of tumor multinucleation is adverse to prognostic of patient survival. For example, if a patient has high number of multinucleation, he or she tend to have shorter survival or more aggressive cancer type. But this study has some limitation. First of all, it was not verified in independent cohort. And secondly, that was based on visual judgment of pathologists that introduce subjectivity problem. I mean, depending on the experience of pathologist, he or she may come up with different conclusion or different decision when quantifying the tumor multinucleation. So we borrowed this multinucleation idea and developed a deep learning driven imaging biomarkers. We term MUNI, that stands for multinucleation index. It's a way of automatically quantifying multinucleation density within epithelial regions. And it was found prognostic of overall survival, disease-free survival, and distance metastasis-free survival among the large multi-institutional cohort. The cohort contains more than 1,000 patients. So this is our pipeline. We built a deep learning model to segment multinucleated cells from H&E images. In the first step, the model was trained on a large data set aiming for segmenting nuclei from H&E images. So there were like more than 300,000 small images. After that, in the second phase, using our multinucleation annotations, we fine-tuned the model to make it discriminate or distinguish multinucleations from other type of cells. So at the end of second phase, that was the second phase of the training. So at the end of this phase, the model output of the model was a mask that contained 3 different colors. Blue for multinucleation, white for other type of cells, and black for background pixel. Once we have a segmentation model to calculate the multinucleation index for a given host light image, what we did is we first extract tiles from big host light images. We use 40x resolution magnification for them. And for each and every tile, we segment the multinucleation and epithelial cells using our model. And then we count the total number of multinucleated cells and the total number of epithelial cells by combining the output mask coming from this tile. And then the ratio of total number of multinucleated cells to total number of epithelial cells defined as multinucleation index for this corresponding host light image. So this is the study cohort. So we verify our biomarker on a large multi-institutional cohort. So we exclude the patients who do not have any clinical data or who were not P16 positive. And we also eliminate the host light images which have poor quality for processing. And after that, we end up having 1,094 oropharyngeal cases from 6 independent institutions. And we use one site as a training data set. So any learning process or any cutoff definition have been performed in the training set. And then we use the remaining 5 sites for the independent validation of the biomarker. So based on MUNI, we examine potential of this index to prognosticate the survival of the patients. So to do so, we first discover a cutoff value in the training set. And then using that cutoff value, we predict each patient in the validation set as high risk or low risk. So for example, if a patient has higher multinucleation index higher than a cutoff value, it was defined as high risk, otherwise low risk. And then we compare the survival curve of high risk and the low risk patient. And Kaplan-Meier analysis revealed that survival of high risk patient is shorter than survival of low-risk patients with significant P and HR values. And we performed the same analysis for individual tumor stage group defined by AGCC 8 edition. And MUNI was found prognostic for stage 1 and stage 3 as well. So as a summary for the MUNI, we proposed a new AI-driven imaging biomarker to prognosticate the output, survival of the patients. It just rely on H&E stained images, which is routinely obtained in the clinics. So this is cost efficient, and this is also reproducible and rapid. So as a next step, we selectively focus on key subgroups by using their multinucleation index. So namely African American and Caucasian Americans. So we compare the tumor morphology of African Americans and Caucasian Americans in terms of their MUNI. So when we look at the distribution of MUNI in African American and Caucasian American, you can see that mean of African American group is higher than mean of Caucasian American. And that difference between the means is statistically significant when we apply Wilcoxon rank sum test. So this finding may suggest us that African American patients tend to have larger multinucleation index compared to Caucasian American ones. And as in another experiment, we also look at the survival of African American patients and the Caucasian American patients in our data set. And Kaplan-Meier analysis revealed that survival of CA group is shorter than survival of African American groups, and that is statistically significant. And these two findings may suggest us, these two findings that are high frequency of multinucleations event in the African American patients and their poor survival. These findings may suggest us that the difference could be related to not only their socio-economic status, but also a more aggressive phenotype in the tumor, in this cancer type. So this is an exploratory study. We assess the population-specific differences by quantifying the frequency of multinucleations obtained from the routinely acquired H&E images. Even though these findings suggest us using population-specific cutoff or population-specific prognostic model, we don't have a sufficient number of African American cases. So because of that, we are not able to create a more reliable prognostic model specifically for African American patients. But that will be one of our future steps. We will try to get more cases for African American group. And once we implement a population-specific biomarker, along with the latest staging guideline, this biomarker could pave the way for developing more personalized and more effective treatment strategies for these people. And this study was just one example of providing some biological insight into differences in tumor morphology between African American and Caucasian American patients in the context of HPV-related cancer. Yeah, that was the end of my talk.
Thanks, John. Thanks, Haojia. So as we've seen from the talks of John and Haojia, that computational biomarkers from digitized H&E pathology showed significant differences between population groups, between South Asian and North American women in the context of breast cancer, and between African Americans and Caucasians in the context of oropharyngeal squamous cell cancer. So, I mean, these are still very exploratory works. Obviously, we still need to validate our methods, train them on multi-institutional cohorts, and then we also would like to correlate these with some of the established genetic and molecular markers. But nevertheless, these are some, these provide evidence, preliminary evidence to the fact that, you know, there are phenotypic differences between population groups that need to be accounted for to build more effective prognostic and predictive models. With that, we conclude our talk, and we are happy to take any questions for discussion. I see that we already have a few questions. Piyush leading with. We'll try to answer those and discuss around them. And in the meantime, probably the others will have more time to think and pose their questions. So Piyush says, since population characteristics are so variable, how will we create models that will generalize and scale? Do we have to change our approach? Haojia, do you have any thoughts?
I think my thought would be like we don't necessarily have to change our approach, but when we like train our model, we should, in order to enable the model to be generalizable across the different population, we have to include some data points from that population to train the model. So yeah, that is important.
Yeah, so I think exactly on the same note, what I think is we may not necessarily need to change the models as such, but in the way we train our models, if we incorporate data points from that specific cohort, then that'll make it more personalized and adapted to them. So we don't necessarily have to change our approach, but train, use our training set properly to train these models. John, do you have any thoughts on that?
Yeah, I think I agree with you. So especially for the deep learning models, which are data hungry approaches, if your training set represent the real world population, then I think we don't need to go with more advanced techniques. So as long as we have well-defined training data that represent the population, I think deep learning model should be enough to capture the signal from the images.
Thank you. Yes, Piyush has a hand there.
Yeah, just want to clarify that, because I think in a global world, especially when you take in context of US, where the populations are more and more mixed. So this is just our way of defining populations, where it's African American, and then there are Caucasian, and then there are Asian populations. But if you see how the world is evolving, even these definitions are getting into more mixed populations, especially in many parts of the world.
Right.
So and then the general expectation from clinicians and many other groups is that we should be able to take a model and be able to scale. So are we going to look into all these different population characteristics all the time, or do we have to change our approach where we say that these variations exist, and we can't go after defining each and every population and the impact of there, but we have to take a different approach? I'm just trying to think aloud, because these are likely to only grow. And you think about the impact of the social determinants of health, where in your personally me, I'm an immigrant to US, so my initial life was in India, where the social determinants of health were different, and then I'm here, which impacts a later time period of my life. And these are all getting mixed together. So these are not very clearly cut off or defined in many of the populations. So it's just a question that how will we overcome these challenges when we expect that these models should perform at scale or for populations?
That's a very nice question. So this is just a very preliminary, very rough, very raw approach is what I would say. That we are taking, but again, the hypothesis for this is coming from genetics again. So what do we have when we have mixed race populations? There's an interplay of several factors. We have the social determinants of health, we have food, we have several different habits, and they're all at play here. So how do we isolate all of these? And even within the race is also not genetically defined. So most of these studies we have taken self-reported African American race or whatever they've reported. So I think that question is very valid to be able to bring this to the clinic and to actually get this up to scale. We need to address all those questions. But this is just beginning as a seed for thought that we need to think along these directions. It's not a one size fits all solution, and we're seeing more and more evidence that in order to improve outcomes, so it's not that if you don't take into account these differences, models don't work. They will certainly work, but in order to refine them, make them more personalized, make them more meaningful, and in also in certain, I mean, this is a very sensitive issue. So especially when we look at prostate cancer, right? So most men are reluctant. We did a survey actually, I think a couple years back, and we heard that most men, African American men, they're reluctant to get diagnosed, to get tested for prostate cancer, and that is one of the contributing factors to why there are such high incidence rates. So if we have a dedicated model which has been tested for them, which has been particularly developed for them, so and if we can communicate that to the community that where there is an specific interest being taken for you, then that will probably incite a cultural shift for them to come forward and get tested. So I mean, again, it has to be presented properly, presented in the right way, in the right context. So again, so these are questions which we want researchers to be thinking along these directions. Probably, there's more evidence that will come from the genetic and molecular sciences community as to how should we be more objective in defining specific populations, and what are the cutoffs, and what happens when there are mixed ethnicities. So I think we definitely need more work there. So this is very raw work, but just to get the discussion started along this. Piyush, any thoughts?
No, I think just highlighting this issue is an extremely important part, and congratulate both the presenters and yourself and the team on doing this, because otherwise we just think that everything is homogeneous, and
Right.
these are very significant issues that do lead to challenges with implementation, because when you start implementing these models, that's when you see that they don't perform as well, and then you're wondering, like, why didn't they perform well? So I think trying to understand them at base and trying to define these data issues at base, I think are just unearthing more of what we have ignored many times in the past. So definitely great work along this.
Thank you. Yeah. And you also had another question. How do you determine the cutoff for the high and low risk? I think this is from John's part of the work. Yeah.
Yes, we just take the average of multinucleation index in the training set. So that was working. So we just want to keep it simple. Go with the average.
It's more heuristic and population based. So we have a distribution, identify the mean, median, then that's where we go for the cutoffs. So obviously we do need to be more objective in determining these cutoffs. But again, as John has qualified it in his talk, that we have very imbalanced data sets, and this is again a starting point. So we'll definitely look for identifying better cutoffs and then also validate this. I think that's where the test of the model is. When you have multi-center validations, if it really works, then that's the test of the model, if it's really working or if it's just garbage.
Yeah, so in my case, I identify the threshold by looking at which threshold give the optimal performance on the training side. So I have a list of different thresholds, and I just plot out the KM curves and see like which KM curve is most separatable.
And then you use the same threshold for the other validation cohort. Salvation.
Yeah.
Thanks, Haojia. Any other questions from the group, from the audience? Well, thank you. Again, I think this is a preliminary direction. There is definitely social determinants of health. So this is something which we are trying to include in our research as well. So looking at the imaging factors and also trying to work with some of the more population scientists who are working on that specific research and put together an integrated and combined model. It's not easy to isolate all these different factors. It's going to be very challenging, but we're trying to at least give some preliminary data so there's more funding, there's more opportunities to explore more in this direction. To you, Piyush.
Yeah, thank you, Rakesh. Well, this was an awesome session, learned a lot. And I think some of this preliminary work is very foundational in shaping the direction for the future. And as we conclude this session, just want to remind, please join us on brainxai.org, join our LinkedIn group where we share a lot of the scientific information. We do these live sessions, so join us for those. We do them monthly. It's easy to register, everything is free. And just like this particular presentation, the prior presentations are posted on YouTube channel. So please subscribe so that you get the latest presentation right at your fingertips. And subscribe our podcast channel called BrainX Talks so that you can listen and share from the wisdom of some of the very experienced professionals in this space trying to apply AI in healthcare. You can always contact me. And thank you, everyone. We will see you in 2022. And this was a great ending to the 2021 BrainX Community Live sessions. Till next year, take care and happy holidays.